Thanks everyone for joining us today for the Pharmaceutical Online live panel discussion covering contamination control strategies for small molecule drug manufacturers. I'm really pleased with the expert panel we have on hand. I'm going to give some quick introductions and then we'll get right into it. Our first panelist, Herman Bosenhart. Herman has 49 years of experience in pharmaceutical, biotechnology, and medical device manufacturing, engineering, and compliance, including extensive industry experience in contamination control. His current consulting work focuses on the areas of aseptic systems, biological manufacturing, and automation computer systems. He's got a BS in chemical engineering and an MS in systems engineering, both from the Polytechnic Institute of Brooklyn, which is now a part of NYU. Herman is president at Bosenhart Consulting and is the author of Commercial Biotechnology, Practical Guidance for Engineering, Operations, and Maintenance. He's the author of over 150 technical papers, a frequent guest author on many forums, and is the co-presenter in Bioprocess Online's Biotech with the Bosenharts. Our next panelist is Liz Broxson. Liz is an aseptic processing and sterility assurance lead in global sterility assurance and microbiology at Takeda, based out of Minneapolis, Minnesota. Liz has a background in benchtop microbiology and immunology, as well as over 10 years of experience in FDA-regulated businesses as a quality assurance and quality control technical professional. In her current role, Liz is responsible for leading the design and implementation of Takeda's global manufacturing sterility assurance program. Liz holds a bachelor's degree in animal sciences and a master's degree in public health, both from Ohio State University. And last but certainly not least, we have Vanessa Vasati Figueroa. She's a chief microbiologist at VVF Science with over 20 years of experience supporting biopharmaceutical companies in sterility assurance, contamination control, and regulatory compliance. She's authored numerous technical publications, holds patents for the Virtuosi training program, and brings global expertise in aseptic processing and quality systems. She holds bachelor's and master's degrees in molecular biology and microbiology. Thank you all so much for being here today with me. I really appreciate it.
Yeah, thanks for having us.
Okay, yep, thank you.
I thought a good way to start off this discussion might be to start with the type of contaminants that we're trying to prevent from getting into our systems. There are three major groups of contaminants. There are physical substances, chemical contaminants, and microbioles. And I'm curious which are kind of most relevant to small molecule manufacturing, or all three contaminant groups are relevant to small molecule manufacturing. Who could I start with on that one? Herman, how about you? Let's start it off with you.
Geez, I was hoping that Liz was going to start with that. Well, they're all actually relevant. I mean, the fact of the matter is, if you look at any types of contaminants in the physical process, you got to just step back and look at what's in the physical process. So there could be anything from physical substances such as breaking off of rouge or rust or whatever within inside of that, or breaking off of gaskets or materials of seals or whatever. So there's some physical ones. In the chemical contaminants, of course, there's, besides the traditional leaching and extractables, there are a lot of other sorts of chemicals that can contain something that's deleterious to the process. And of course, the microbials, which is what we've really been struggling with over the years, trying to free ourselves with. And those can be anything from molds to yeast to bacterias and so on. And so we see a wide variety. If the facility is relatively in good shape and relatively new, we tend to focus ourselves on the microbial contaminants. If the facility has been around for 10, 20, 30 years, we start to see some physical degradation of seals and gaskets. So you start to see some of the physical contaminants. And if we are starting with some new materials, some new processes, some new chemicals, some new formulations, we could start seeing a little bit of the chemical contaminants. So I'll kind of then throw it to the other folks out there to see if they want to add to that.
Yeah, well, let me ask this. We're talking broadly about contamination control strategies for manufacturers today. I mean, do these different types of contaminants require separate, individual contamination control strategies, or is there a comprehensive strategy, that we can talk about that will cover all three?
Yeah, I think I can take this. I can at least start it. So all that Herman shared is in fact correct, but I think to go double down on the controls, they come from not just the facility, but also the process. So for small molecules, chemical contamination or cross-contamination for APIs, anything that can, what we call in our legal code here in the States, adulterate the product is considered a contaminant, right? That adulteration changes the intent of your formulation and how you deliver that product ultimately to the patient, right? So that can come in the way of particulate, right? Many different types of particulate, from packaging materials, from rouging on equipment, poor cleaning processes. When you are thinking about the types of contamination and pulling together a strategy, you can do it a bunch of different ways. I would recommend that they do be included together sort of holistically. You need to understand the types of contaminants, and microbial also has many different classes. So viral is another type of contamination that, depending on your process, some cell and gene therapy, you need to consider viral contamination too. So we should address all types of contamination, your CCS. You should understand the control measures and how they function to remove a specific type of contamination. Not just say it removes contamination. Does your cleaning program specifically address chemical forms of contamination, right? As well as microbial, and you understanding which of those, certain types of cleaning agents function in a way to remove microbial, which types of microbes, etc. So yes, it should include all three, but they should be strategic in the way that you describe the contaminants, when and where they enter your process, and what are the specific measures that address the specific type of contaminant. So hopefully that helps.
No, that's great, and I think that sets us up well for our next section that we want to cover here, which is architectural layout. The way that a manufacturing site is designed. And Liz, I promise I'm going to come back to you shortly, but I want to get Herman in on this one, which is, what are the fundamental principles of good architectural layout for contamination control in small molecule manufacturing, Herman?
Yeah, I mean, the first, and I'll use a general word, and then we'll work our way down for there. Is we have to prevent contamination from people, from external materials, from equipment moving around, whatever. So the first thing is unidirectional flow. That's the most important thing. There should be a singular path as they come in and go out. So operators should be able to go in, gown up, step through the different grades, do their process, and step out in a completely separate area. And quite frankly, you should have 180 degrees between entry and exit. The flows for people as well as material airlocks. Material airlocks should also be 180 degrees away from each other, and there should be an entrance in, a material airlock in, and material airlock out. Each way, that basically controls the flow. Now, the other issue that we've been doing, and we've been adding to it over the last 10, 15 years, is to make sure we have a clean corridor for entry, and we have a dirty corridor for exit. So when we leave, we don't contaminate our corridors as we go in and out. That's why I mentioned that 180 degrees. So you have a clean corridor, clean materials, clean people, just gown, going in and following a particular path. And on the way out, there is a path for waste, which does not cross over the people leaving and materials leaving. Waste has really got to be a very, very separate area and has to go to whatever kind of decontamination mechanism or collection and destruction it has to be. But those are the basic fundamentals. Grade changes, we should be an airlock between every grade change. We're not going to have just gang them all together and keep our fingers crossed. So there's a number of things that we really have to do in terms of how we proceed from I'll just say from the gowning up process all the way through to the exit process. And there's no backward motion in this. You move the materials through. Clean materials come in, they're used, and then they're moved out into the waste area. People move out. Pallets move out if you do moving pallets around. And today what we're looking at architecturally is to be able to put a containment area for pallets within the different zones so that we don't bring it out to a lower classification. So, I mean, if you want to really look at it from the standpoint is unidirectional flow, airlock between every grade change, clean corridor and dirty corridor as kind of a summary.
And well, pardon my ignorance on this, Herman. When you say 180 degrees in terms of a separation, is that suggesting a distance? What does that mean exactly?
That so they don't overlap. But a lot of times people want to cheat, so they put the personal airlock in, and then they put the personal airlock out next to each other. So you have this one corridor where the kind of stuff kind of mixes around, and people will mill around, and there's a temptation to open doors or whatever. So you have, so on one side of the building, you have the in, and the other side of the process area, you have the out.
Liz, I might ask you this next question, which is how do regulatory requirements inform layout, the kind of layout and design that Herman is talking about here, including those exact elements, airlocks between grade changes, unidirectional flow, separate clean and dirty return corridors. Is that cleanly laid out in regulatory documents? Is it something you have to document?
Yeah, so there are a lot of descriptions of the expectations for the buildings in which you're manufacturing laid out in the various regulations. With the updates to Annex 1, there are some pretty specific requirements, especially around airlocks and when you can have shared airlocks, when you shouldn't have shared airlocks, what are the requirements for the air quality of those airlocks, things like that. And I think that we're seeing really most importantly, a trend in regulators being really unwilling to accept poor facility design or aging facility design as an excuse for not complying with those things. Where, previously in years past, we could kind of risk assess or justify our way around having some of these not ideal designs. I think we're seeing a trend where the tolerance for that is really decreasing.
Yeah, well, I mean, the point of the shared airlocks is really bad. I've seen in the past where people and materials share airlocks. You can't do that. And even today, the personal airlock coming in, personal airlock coming out, controlling that, managing that is not good. And it never really works out very, very well because ultimately you'll have people degowning and gowning in the same area. And that's just unacceptable.
Yeah, so I imagine this has to be kind of thought about in the very earliest stages because, you've mentioned aging. Liz, you mentioned kind of aging facilities. At some point you can't really redesign, a wholesale a facility, right? It's got to be kind of set up from the beginning. I mean, I'm just thinking you've both named a couple of common mistakes that people made with respect to architectural design. Or are there any, I guess, final just sort of mistakes that you would mention that maybe that you've seen in practice or that you would, caution everyone who's listening to this webcast to avoid making?
Yeah, I'm going to say the biggest mistake I think people make when designing a facility is underestimating the square footage or the actual space you need for supplying clean air and removing dirty air in material airlocks and gowning rooms. Those are often like tunnel vision type of rooms that you think people are just passing through, but also balance that with like the bigger the space doesn't necessarily mean the more people you put in it, right? So we need to understand that, when you're designing a facility, having adequate space for people to come in, have a place for materials, have a place for cleaning agents so they can wipe down certain materials, and have the appropriate flow that comes with you understanding not just flow, but having the sufficient physical space. Because, if I had $1 every time I saw an EM excursion from a gowning room, I would be a trillionaire, because you go to every facility, all their trends, they're like, oh, they're in the gowning rooms. Well, that makes sense. People are there. Well, no, that's not just the reason. You should design your gowning spaces and your material transfer rooms to have adequate air exchanges, to have adequate airflow, and to be able to host the equipment that you need in the room to do the work in that room. And that's like one of the biggest mistakes I see people make, both in retrofitting clean rooms, right? And then you've got these storage rooms that are like huge and nothing's in them. And so it just, borrow, if you're trying to retrofit an aging facility, borrow or reconstruct space from another part of the facility. Or if you're doing a new design, when you think you have enough space, add more. That's my recommendation.
I want to bring a question in from the audience that I think is relevant to this discussion. Please continue sending questions in, everyone who's listening. Also, I failed to mention this at the beginning, but all of our panelists have expressed an openness to answering questions online, either via email or on LinkedIn. So if we don't get to your question today, please feel free to reach out to our panelists. The question that I'm going to bring in right now is what are the mitigation plans in the case of PAL in and out, if those are shared?
All right. First of all, I'm just going to tell you, you shouldn't be sharing them. Okay. So that's the bottom line. Now, there are mitigations. So here's what you can do. There's two things. One is increase your ACR, the air change rate on the in that particular room, and bringing up the air change rate. Secondly, is one of the things we found in the older facilities is where the air supply is and the returns. The air supply should be obviously on the ceiling, but the return should not be mid-wall or even on the ceiling. It should be flush with the floor. Secondly, is to control the traffic of the people, to putting timers on the airlocks. So if I'm gowning up, Ben and I are going to gown up. We go in, we open up the door, we close the door. We put on our gowns and whatever, and then we step over and we step across. That particular airlock should have at least 3 full air changes on volume before anybody's allowed to go in again, whether they're gowning up or gowning out. So if somebody's outside, there should be a flow control so that if you see somebody in there gowning up, you're not going to be gowning out. So you're going to have some interlocking controls, some entry and exit controls, and whatever. In addition to that, the PALs specifically need to have their cleaning procedure updated to increase the frequency of cleaning. And I don't mean just the floor. I really like, just the floor. No, floors, the walls, and the ceiling. You start from the ceiling, you work your way down. So higher ACR, higher frequency of cleaning, making sure you have flow control of people going in and going out, and make sure that people adhere to the practices of correct gowning and don't be, playing with their shoes and then stepping into the Class C room, that they make sure they wipe their hands or whatever. And in most cases, I tell people, put cameras in the gowning rooms so to make sure that people are following the gowning procedures.
And I'll just add, making sure all those controls are proceduralized and making sure that the training is robust. And one thing that we've really been focusing on is making sure that everyone understands the why behind these things that are in the procedures, because it's annoying, right? Like, I don't want to wait 5 minutes I'm ready to go. I'm ready for my lunch. I'm just going to go in there. No one's in there. It's not a big deal. But understanding, hey, this is a lot of particle generation and potential microbe contamination, and the system needs time to clean the air. And here's all the reasons why it's so important to follow the procedure. It really makes a big impact for folks. And folks don't inherently want to do the wrong thing or take shortcuts or things like that. But in the absence of having the information of why this is important, they could make a poorly informed decision.
Yeah, I mean, I've seen situations where people they prop open the door on the airlock to let folks in and out. Now, that's why maglocks and timing systems and so on are really critical.
But I think behind both of the examples you guys just shared, often is production pressure, scheduling pressure. And a lot of people don't understand, if you've got an aging facility that is cranking out a blockbuster drug product, you have to take into consideration the total time that it takes for your production crew to get inside to the core, right? If we're talking about a sterile facility, a sterile manufacturing facility. Because if you've got 8 people who support that process, and each person takes 10, 15 minutes, and then you've got these air exchange requirements in between, maybe it takes you an hour or 2 to get in. And then on a single shift, you've only got 6 hours of production time out of people, and before you need to take breaks and shift changes and stuff like that. So if you are quality people trying to implement these systems and temporal controls, then you also have to work with production to understand realistic scheduling, because the scheduling will always put the pressure on people, and people will always then be forced to take shortcuts because they think it's coming from leadership. So there's a really important balance there. Yeah.
Very true. Very true.
I want to move into HVAC systems next. We had a question from the audience about mitigation strategy for facility design of recirculating air from HVAC systems. What beyond just general air conditioning, what are the critical roles of pressurization, segregation, isolation in HVAC systems for preventing contamination?
That's the lead in. I mean, the first thing is you got to, there's 3 aspects. One is pressurization, making sure that you deal with the pressurization to make sure that the contaminations will be blown out or be captured by the HVAC system. So you may be the traditional one where the center core is pressurizing, then it goes to the airlocks, and then goes out gradually. But more and more, and especially in the small molecule area, and dealing with a lot of the even new biologicals, you now have potent compounds, you have very, very active compounds. So you have to use pressurization from a standpoint of sometimes implementing bubbles and sinks. A bubble is air blowing out from a particular room. A sink is air blowing into it. So say you may have a potent compound as a small molecule. Right next to that would be an air sink to absorb everything. And then on the outside of that would be an air bubble to make sure you pressurize the corridor and pressurizing that particular process suite. So pressurization is 1. 2 is what we call is process isolation. So that the one process train is, from an HVAC standpoint, is all one unit. It's one single unit, and it's controlled all within that. And what we've been doing more and more lately is HEPA in and HEPA out. That controls what's going on in that particular suite. So if you have a biological, you have a potent compound, you have some kind of super active compound, you HEPA air all the air return, as well as supply HEPA in. And then the last one is segregation, is that we should be using smaller and smaller HVAC units only for that particular process area and that particular process train. Don't have one of these like the old legacy systems where you had one gigantic HVAC and it serves 5 or 6 processes and 25 rooms or whatever. Those days are over. They're too expensive. They cost too much to maintain. And if the HVAC crashes, you're out of luck for a whole bunch of areas. So pressurization in terms of protecting that environment, the pristine environment, and protecting the people in the exterior. Segregation from the HVAC standpoint, and process isolation, keeping that all in one particular envelope is really critical.
I want to bring another question in from the audience now, which is it necessary to get a high air change rate? High value may be more than enough with energy wasted. I've seen some companies reduce ACR below the value cited in FDA guidance, ISPE, but still achieve satisfying results. What is your opinion about this? Anyone have an opinion on that air change rate?
You want to throw the dice? Throw the dice. I wouldn't do that. There's a reason for that. There's a reason, and yes, you can achieve a Class C environment at an ACR of 15 to 20, and you can get a Class D at 10. But the reality is it's you have a tremendous amount of risk associated with it. And how do you have it asymmetrically covering the area? Do you have a floor level return? The reality is I would not go. I have if you want to look Class D, I'd use 15 For Class C, I tend to use 25 to 30, and also do a risk assessment. What are we doing inside? If we're handling media for prepping, or we're handling some kind of potent compounds or whatever, I usually boost the air rate up to make sure that I have the level of capture to protect the process as well as the people. So you've got to look at it. You're saving energy. How much energy are you really saving, and putting what at risk? The product at risk, the people at risk, the environment at risk. I mean, I think that's really foolish.
Are there any new HVAC technologies, new or emerging, that offer advantages over traditional large air handlers? I guess particularly for containment and reliability.
Fan wall technology. People are starting to use more and more fan wall, where you have multiple small drives in the HVAC as opposed to one. So you could have an individual failure. You could, again, it's to risk failures.
All right, excellent. I'm going to move on from HVAC and talk for a few minutes about single-use disposables. How do single-use disposables? Well, first, maybe we can just get a definition of how single-use disposables are used, and I'll toss that one out to the group and see who wants to tell us what a single-use disposable is.
Yeah, I'm happy to address it. Yeah. So single-use and disposable together can mean a lot of different things, and I think it depends on your process. So I'm seeing a huge trend towards single-use, like liners, bag liners for vessels, which are very helpful. There's a lot of different types of technology out there. I'm by no means an expert in single-use technology, but single-use filtration lines, single-use materials, even down to pre-packaged, pre-sterilized, single-use, obviously, like small containers for sampling. So I think that there's a lot of different ways on the line that you can incorporate single-use materials. They help to reduce cleaning and sterilization, and that's why they've become so popular, right? So you don't have to have, you have to have different measures in place if you're using a liner, a single-use disposable liner in a production vessel, a formulation vessel, than you would to actually clean the vessel if you're doing it using the vessel itself, stainless steel, right? So the degree of cleaning, the cleaning validation requirements shift. Sometimes they reduce, right? And so there are pros and cons for that. There are things that go wrong with single-use liners and bags and stuff like that. A lot of people are using single-use bags to hold the final material that is then tubed into the production vessel, right, itself. And so you can go right into sterile filtration, and then the whole line gets disposed of without having cross-contamination, without having hard pipelined aseptic connections. And so there's a lot of different advantages. But then the types of controls you need to put in place are a bit more upstream. So incoming inspection, obviously you need to supply, get good qualified suppliers and manage those suppliers. Not all suppliers are created equal. Not all sterilization cycles on single-use materials are created equal. You see some of them that they'll reprocess materials and then they degrade before you even use them. I mean, there's a lot of things to consider when you're trying to implement single-use, right? You can't just say, oh, this sounds good, I'm gonna go to it. You need to understand where you're using it, how it affects the product, what are the extractables, leachables, or what happens from the materials to your product, how they interact. There's a lot of different things that you need to consider even when you're doing aseptic connections of the fill line to production, because you need to validate that. You need to understand how those are being used. The throughput, the flow, everything changes when you're starting to implement a totally new system. And then waste is also a big issue. So, it does kill the little eco-friendly, soul that I have deep down inside as we see the whole industry go from what I thought was fantastic, strong, high quality grade stainless steel vessels and pipelines and all of that, shifting towards plastics that contain chemicals, forever chemicals, blah, 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 but also that get put into earth and wasted. So, there's a lot of sustainability conversations happening around this, but there are advantages, and I see a shift within industry going towards implementing these as effective contamination controls. They are, quite effective when done properly. So just some things to think about, when you're implementing them. And then the last thing I'll mention, all the failure modes, all the things that can go wrong with bags, with double liners, with, final product bags, with sterile filter lines, operator errors, what happens when they get dropped. I mean, there's a lot that can happen that goes wrong with these things. Don't underestimate implementing those types of controls as well on your CCS.
Yeah, we're at a particular stage where we're even on the fill finish side, all the way from the surge tank, which is a single-use bag, all the way to the tubing, and the filling nozzles are all single-use. So when the fill is over, you take everything and you throw it out and you reline and you put it all back together again.
Yeah.
And we've gone TFF, the tangential flow filtration, besides the containers and bags, all. But here's a very practical one that I'd like to bring out. What about all the vents? What about all the air filters, all the nitrogen filters, all the gases that we do? We still have people out there with filter housings and cartridges or whatever, decorating the whole facility. Using a single-use disposable just on a vent filter, just on an air filter or a nitrogen filter, makes all the sense in the world. And you could see it, it's in place, whatever. So that was one thing I'd want to encourage you. But I want to go back to Vanessa, and I'm going to ask you, that whole question before of running the ACR down in the HVAC below the ISP, FDA, and the ASHRAE design, that means that we're not running that particular room, suite, or whatever at the same conditions. I don't know. That doesn't make me feel good. Tell me more. Say something.
Well, I don't know where the question's coming from the audience and why they would want to do that. I think putting the controls in place to make sure that that room is functioning on its own and different from the other parts of the facility, I don't know if that's even worth the effort. But I think, a lot of my experience comes from supporting, sterile injectables, other small molecule cell and gene therapy. I can imagine, Herman, that there are facilities out there for different types of product, non-sterile products, in which that could be okay, depending on the room and its condition. But I wouldn't suggest it for anything that's related to production, formulation, those kinds of rooms. So I'm not sure. As an industry, I think, with understanding risk management principles, a lot of people sometimes take them too far and think, well, if it makes scientific sense, then, maybe I don't understand, what could go wrong yet, and I could give it a shot and then see what goes wrong. That's not a risk that I'm willing to take. And I don't know and I don't know if it's worth the time and effort for one room to be set different air exchange rates than another. But, to each their own. And if they can defend it and support it and not get inspected to it, then, I think it could make sense for certain products, but I don't think it makes sense for a majority of the pharma products that I'm comfortable with.
Okay, good.
Vanessa, I want to stick with you to talk a little bit about barrier systems and isolators for small molecule drug manufacturing. Maybe you could tell us what role barrier systems play and perhaps how restricted access barrier systems and isolators are used effectively.
Yeah, so I love barrier systems. I think that if you read EU Annex 1, you see, if you read between the lines, if you understand, the intent, as well as the plain language that's in the guidance, it's very clear that barrier systems is where you need to be. So if you're constructing new facilities, the expectation is you have them. And you have isolators is obviously the preference if it makes sense to build your process in an isolator. I think also the expectation for older lines or aging facilities is that you do a retrofit for a barrier system that makes sense to your filling line. And the whole principle is very simple. It's to provide a barrier, a separation between people, the operators, and the product. It's a physical layer of protection to reduce particulate, chemical, air, whatever contaminations coming from the operator, the people, right? Number one in the room, but also the background area. So for small molecules, for these types of products, I think it depends on the type of small molecule you have. So barrier systems can be for containment purposes, right? Especially if you've got powders or if you're compounding APIs or something like that. They can help contain while also simultaneously protecting, obviously, the operator, but cleanliness of that, that compounding step, if you will. When you're talking towards fill finish, barrier systems really provide a microbial contamination control unlike anything else. So for isolators, it allows you to decontaminate the area, right? Using a validated process. Even some closed RAB systems, there's new technology out there that's also letting you decontaminate them. I haven't worked with it yet, but it's really encouraging and exciting. But even closed RABs or even open RABs, you can clean those systems, let the area recover prior to doing your actual fill finish. So they give you a lot of protection for the product. I think that's why we're in this business, is to deliver products to patients, right? Not just to formulate, but they reduce contamination risks. I think that they're an expectation from regulators, but they play a huge role and a different type of role depending on the product and the process that you have. Yeah.
What about the differences between, say, open versus closed RABs, open versus closed isolators? Are there differing levels of sterility assurance associated with those different systems?
100%. I always say, we just actually gave an entire webinar on this concept. You can look at it as like a spectrum, okay? And it's important that you understand that not all barrier systems are created equal, okay? And all barrier systems have vulnerabilities in them, okay? So open RABs are just like that. They're open. They should be opened only under very rare or specific conditions in which the line otherwise wasn't retrofitted or wasn't designed properly, and you have to get equipment in or you have to do a specific intervention. Under those conditions, it does provide sterility assurance, but it's not to the same degree as some of the more closed systems, right? It's quite logical. And while you're implementing those controls, if you do have to open the RABs, you're isolating product. You're probably, quarantining a certain portion of the product because you're opening it, exposing it to a background condition. If you go towards closed RABs, that's even better. That means the door is never open and you're operating strictly through glove ports. But understanding glove ports are not perfect. They're not, things do go wrong. Leaks, pinhole leaks, they're not installed properly or they're not disinfected properly. That's really important. And so, while you don't have people's gloved and garment hands in them, you do have them through glove ports, and those need to be managed properly. And obviously the aseptic interventions need to be done properly as well. Just because a lot of people think, oh, well, it's a sterile glove. It's not. It's not sterile. The area inside of RABs is not sterile. The area inside an isolator is not sterile, okay? It can be sterilized, and at a moment in time, you can say everything in there is surface sterilized. But the moment you introduce materials, air, it's no longer sterile. And then as you go from a closed RABs to an isolator, isolators have glove ports too. The newest technology with the greatest degree of sterility assurance are gloveless isolators. So you can see that if you go from open RABs to closed RABs to isolators to gloveless isolators, you get an increase in sterility assurance, and you have better controls, and you reduce the impact of surrounding, operators, bacteria coming from people, gowning and stuff like that. So I think it's important to know what type of barrier system you have, how you can decontaminate, and how you should perform aseptic operations in there. And then, we don't have enough time, but we can talk about robotics too. They give you a whole different level of, control in the way that you are designing the intervention to minimize, airflow disruptions and movements and slow and deliberate, these types of principles. So lots to be said about barrier systems. I think I've become like a fan of barrier systems, talking about them in my recent years. So.
But there's one aspect, a lot of folks have old lines. They put a open RABs around them and they say, oh, we're good. And what we've seen over the years is then their air is being supplied by the room, and they don't go all the way up to the HEPA filters in the room. You've got to go and made up to that. But the other big issue is the HVAC in the room controls the contours of the air within inside that RABs. Mm-hmm. And part of the problem is we've seen cases time after time where the airflow in the room there are 3 returns on one side and 1 on this side, and the air is being pulled in a particular direction. And what happens is you have half of the line or half of that area is not even being covered by the clean air. We've seen situations where the air is out of diagonal and actually generates a swirling vortex in the middle of the RABs and actually suspends particulates up and holds them in the RABs and doesn't remove them. So if you're going to look at an old line and you're going to, just throw RABs on top of it, you have to completely look at the HVAC in that particular room. You've got to do a computational flow dynamic model of the air inside that and really understand, do you have the HVAC coverage? So I think that's one of the most important things that we've come across, and we've seen a lot of fallacies with, open RABs or even RABs that are active, but the bottom end of them are open. They have the separate air, but if the bottoms are open, it's very much dependent upon the HVAC in the room and the opening and closing of doors. So we really got to focus ourselves on close active RABs. We got to go to isolators, and I'm a big fan, and I've bought a couple of the gloveless isolators, and they're the best.
Yeah, I think can we address kind of question? Oh, go ahead, Liz.
Oh, sorry, Vanessa. I was just going to say, I think what both Vanessa and Herman are saying is, understanding the limitations of these barrier systems. And again, my favorite thing is training and making sure that people understand the why, and having the right information to be able to make the right decisions for behavior and actions. So something like an airflow visualization study, which Annex 1 references as being part of the training program for any aseptic operators, so that you do know what the air is doing inside that barrier system. And your controls are, the amount of control that you can get from a barrier system is only as good as your inputs, whether that's something like the development of a VHP cycle or something like training to make sure that when your operators are physically putting, their gloved hands into a barrier system, they understand what they should be doing. And if you have a really, really, really great isolator design and a perfect VHP cycle, but your operators aren't behaving the right way with that isolator, it's not a magic box, right? And I think a lot of people are sometimes like, oh, it's the isolator, it's VHP'd, everything is sterile, and that's just not true.
Yeah, and because operators don't consistently deal with inside the isolator, so they'll pile up tools, they'll pile up disposables, they'll have their EM plates will be piled up there, they'll tear, and they'll pile up all sorts of stuff all over, sometimes obstructing air, just sometimes not being very tidy in there, and they don't dump it off into the dump spot in the isolator. So there's a lot of watching and understanding of the behaviors inside the isolator. And the other thing is, before the visualization studies, rather than burning all the visualization studies and putting all that time, do a CFD, do a computational flow dynamic model. You put it on your computer, run it, and if it makes sense, great, then do the visualization. But don't just go ahead and just say, oh, I think it'll be just fine, and go ahead and fail the visualizations.
Yeah, I think that what the take home from this conversation is that not all barrier systems are created equal. The barrier system that you have for your product filling line needs to be fit for purpose, for your purpose, right? It needs to consider how you operate. And let me tell you, if Herman, like how he described, if you've got buildup of tools, or if you've got glove ports that are in the wrong position, or when they're in rest, they're touching a line or something like that, that's a design issue.
Correct.
I can't fix that in my CCS, okay? So that's a redesign issue. That's working from a microbiologist along with an engineer to change the position of the glove port. You do that during a shutdown. Don't just deal with it, right? You've got to make those changes, because small changes give you big yields when it comes to, grade A conditions. And I think that we underestimate the value in working with engineers to get redesigns done, even on isolators. Yes, they cost money, but you know what? We learn more about the process when we run it 100 times as opposed to just some of the design spec conversations initially. I wanted to address, Ben, if it's okay, that one question, because it's such a hot topic on stopper bowl placement for isolators.
Sure.
Can we do that one? Yeah, so Liz, maybe you want to add, but let me read off the question. I think the question was, is it required to sterilize stopper bowls for isolators, or is VHP sufficient? I have a lot of opinions on this. I think that the industry, first of all, it depends on the type of isolator you have, right? It depends on if you've got transfer ports, it depends on the type of stopper bowl you have. But it's my recommendation that, yes, you sterilize the stopper bowl, because stopper bowls are notoriously configured in a way that there are crevices, just vulnerabilities that VPHP perhaps cannot reach, right? VPHP is a surface sterilant, okay? And yes, it can reach most surfaces if they are properly exposed. The problem with the physical configuration, the geometry of a stopper bowl, is not everything can be properly exposed, in my experience, at least with the old school stopper bowls that I've worked with. So I think that as a control measure, you should pre-sterilize your stopper bowl in the traditional way that we've been doing it for 20 years, for 100 years, Herman will tell me. And then you have to install that to the line. The next point of hot topic debate in the industry is whether you take the bonnet off or you leave it on during the installation to the isolator, and then you close the isolator door and run the VPHP cycle. I don't know that there's a perfect answer for that. I think it depends on how you do your equipment setup for the line. It depends on your VPHP cycle, and it depends on what you can qualify during that VPHP decontamination cycle. So it's important for you to think about that. But yes, I recommend you pre-sterilize a stopper bowl and then install it. That improves your odds for decreasing microbial contamination, but also particulate, because you're gonna have to do an equipment rinse, right? So some things to think about.
Yeah, Vanessa, you're absolutely right. Think about the mounting systems that the stopper bowl goes through, and how you have to place it down in there. I mean, I've seen stuff with bolts and nuts and pointy things. You have
people's bottoms all over them, like they're literally physically, the stopper bowl's 50 pounds. I mean, it's like it's an Olympic feat to get it installed properly.
I know.
Yeah.
Yeah. I want to move on to material transfer and Annex 1. And Liz, I'm going to come to you on this one. And it ties in a little bit to a question that has recently come in that's about, using an older facility. Controls have been working for a while, however, may not be in par with new regulations that are coming out. Is it possible to justify old practices with appropriate risk assessments and supporting data? Just, with that as some additional context, Liz, what is the significance of Annex 1 in formalizing material transfer procedures for contamination control?
Yeah, so this is a little bit of a cheat because that question came from my friend, and Laura, no. But, I think Annex 1 has given some really helpful context written down to say, why is the process of material transfer so important, and what are the possible repercussions? It's listed as one of the main sources of contamination in the clean room behind humans. And they've been pretty specific about all the which ways you can transfer materials and how it should be validated. So I can say at Takeda, we have taken a lot of care to go into the disinfectant efficacy requirements around material transfer and the validation requirements. And we take a risk-based approach to each of those. So for example, we wouldn't say that you have to perform disinfectant efficacy or material transfer validation for every single item that gets transferred, but using a risk-based approach to say, here's all my materials, I'm going to choose my worst case materials that aren't already covered in a disinfectant efficacy study and challenge those specific materials to make sure that the disinfectants that I'm using for a manual material transfer, so I'm not talking about something like an autoclave or a rapid transfer port or like a VHP transfer chamber, but a manual material transfer. And make sure that my disinfectants are actually efficacious against those materials that are being transferred. And then for our critical areas, our grade A and grade B areas, those are the ones that require the validation of the transfer process itself, which is like a bioburden type study to ensure that you have an acceptably low bioburden on your materials that are being transferred. But I think really having the regulation there to spell out, this is a high risk and these are the implications of you not controlling that risk. And now we want you to prove to us that you have controlled that risk. And then it's up to you to address that and document in your contamination control strategy and in your supporting procedures and protocols and reports that that control is there.
That's excellent, Liz. Does anyone have anything to add kind of on Annex 1 and perhaps, new requirements or changes that perhaps folks that have been working, in manufacturing for a while may not be aware of, but should be?
Yeah,
well, I'm going to suggest. Go ahead, Vanessa.
Yeah, I think the fact that you need to now have a documented material transfer strategy. A lot of companies did that because it was a quality system, good thing to do. But now it's explicit in Annex 1, and a lot of people don't really realize. Along with that documentation and your risk rationale, like what I talked about this sterility assurance spectrum, there is, you need to have a program that goes and understands the level of risk from your grade D areas or your uncontrolled, non-controlled, the cleaned areas to the grade D, to the C, to the B, to the A. You need to have it documented, a list of materials and the method by which they're transferred. Like actually document that. And that needs to be a standalone document. It doesn't go in your CCS to the degree of detail that you need it to defend your rationale in front of a regulator or to just support your process. So you need to have a standalone document that describes your material transfer program and how the things that are critical get either sterilized or they're sterile supplied and transferred using VPHP, right? That's important. If and when it can't, there should be very rare exceptions to that rule. That's an EU Annex 1 rule. If and where it can't, you need to make sure that material absolutely needs to go in the critical space. And what are the controls you put in place to make sure that it's not contaminated, right? And so having that documented thought rationale, the program in place, the required materials, but also a list. You can't just let laptops go through, guys. You can't just let a vendor bring in any old thing. You need to know what's going in your clean room, where, and how it's being, reduced from one specific grade area to the next. And so I think that's new. That explicit expectation written down is what Annex 1 is talking about, right? And now, once you have that program in place, you then describe it in your CCS, okay? But your CCS isn't the only place that information should exist.
I want to end. We're running short on time here, and I want to get to just maybe some thoughts about training for operators and technicians. But before we do, while we're on the subject of regulations, are there any updates or key points to consider from industry bodies like PDA regarding cleaning and disinfection guidelines? I think they had a new guidance that is either just come out or is soon to be published. Is there anything that folks should watch out for there?
Yeah, PDA Tech Report 70 is actually in the process of being revised. So I'm part of the team that's working to revise that. So that's very exciting. But definitely, yeah, we're looking at, as part of that revision effort, we're looking a lot at industry trends, and USP 1072 is out for commenting, I think, as well. So definitely there are industry documents that are being revised around cleaning and disinfection.
Yeah, there's also
Excellent.
a new one. There's a new one, a brand new TR, not a revised one, but a brand new TR from PDA. I'm on the committee for that, where we will describe these expectations for transfer from grade D to C to B to A, the list of materials that you need. So there's a really great expert level, passionate group of people that are developing that TR. It won't be out probably for another year or so. if you understand the process, but all the technical, input that goes into something like that. So I think PDA 70 is great. It's under revision, and then there will be a brand new technical report on material transfer processes that'll be coming out. Look, next year, 26, mid to late 26. Yeah.
Just as a one note about material transfer, laptops, cell phones, people, operators, technicians, whatever, wandering in and out. They took the laptop from their desk. They wandered in. They kind of, do a little bit of this, and they wander on. All that's got to be included. What Vanessa was talking about, everything that you may move in needs to be included, and you have to be very strict about it. All the contamination on the laptop from their lunch and everything like that goes in. The other aspect is, if you really want to see what not to do for material transfer, just go to the FDA's 483 for Emergent Biosciences in April 2021. Read that, take a look at that, even with the redaction. Those are things not to do.
Right. All right. We probably only have time for one last question from me. And Liz, I'm going to direct this one to you on operator and technician training. Something you said stuck out to me when we were chatting, in the planning of this panel, which was people are the biggest source of contamination. What are the key challenges and best practices for training operators and technicians effectively in contamination control, including, things like paper gowning and aseptic techniques?
Yeah, I think Vanessa and I have both touched on what I think are two really critical things. And the one is understanding the why, and the other would be understanding things like production pressure. So again, people aren't going to work thinking, I'm going to do a really bad job today and I'm going to feel great about it, but they might be feeling very rushed, and they might, as Vanessa mentioned, have the impression that this is direction from leadership, right? Like, we need to get these batches out the door. I don't care what it takes. Get in there and get it done. And that makes, like, I'm feeling like I have to rush now too. And this is a big scenario. And then, like I said, understanding the why, things like contact time for your disinfectants. There is a reason why your contact time is what it is. It's because you validated it to be that contact time. And in some cases, you may have failed your validation at a lesser contact time. So it's really important that the why is well understood by the operators. And I would also add, using examples that are meaningful to the people that you're training. So I gave a training to some contract cleaning service providers, and they were at a site that has cold rooms. And there's kind of this idea like microbes don't grow in cold temperatures. And so when I was training, we talked about food, and your leftovers aren't good in the fridge forever, right? Like, you get mold on your food in the fridge, or it gets slimy, or it starts to smell bad. Those are microbes. Those are microbes growing in refrigeration temperatures on the food that they like to eat. So even though you might not have, a sandwich or soup or leftover takeout Chinese food in the clean room, it's the same principle, right? And I heard a lot of people afterwards being like, yeah, that's a good point. That is a good point. So having those meaningful examples to connect the why, I think, is really helpful.
Yeah, let me just jump in on the operators. The average operator will do exactly what the operator who has been supervising them will tell them to do. So if they actually tell them not to do the right thing, they will do not the right thing. You have to make sure that the senior operator, the supervisor in there, is very, very aware of what is the correct way to do it and will enforce it. And they also need to know what good looks like. I mean, we had that chat this morning about all the rouging and all this sorts of stuff. I was in a situation where I saw a door had all the black grease on the side of it. They said, well, it leaks like that all the time. Don't worry about it. Well, no, it's no good. So the leadership has to know what good looks like and has to enforce it, because the operators, the common everyday operator, will only follow what the person will do. And they may not even care about what's in the SOP.
Well, we are going to have to leave it there. Unfortunately, we probably could have taken this webcast on for another hour, but I really want to thank my my panelists, Vanessa, Liz, and Herman, for a really fascinating discussion. There was a final question here about what was that 483 reference that highlights improper material transfer. I believe that was emergent biosolutions that Herman mentioned during the rollout of the COVID vaccines. But we will try to address some of your questions that we couldn't get to. Please do reach out to the panelists if you want to speak with them individually. And thanks everyone for your great questions and for attending today. I really appreciate it.